Hi everyone, my name is Novaselska and I'm a first-year master's student presenting my project, Human Embryo Mitochondrial DNA Heteroplasmy Dynamics and Clinical Significance of Mitochondrial DNA Variation in Pre-implantation Embryos from Fertile and Infertile Populations. So before I begin today, I'd first like to thank MITO2i and Thomas Zacco Scholars for the financial support and for this incredible opportunity to present my project to all of you today. So I'll first start off by giving a background of important concepts surrounding my project. So what makes the mitochondria really unique compared to other organelles is that they contain their own genome called the mitochondrial DNA. Mitochondrial DNA is circular and composed of an inner and outer strand, and unlike nuclear DNA, multiple copies of mitochondrial DNA exist within each mitochondria, so a single cell can contain hundreds to thousands of mitochondrial DNA copies. This also means, however, that both normal and mutated mitochondrial DNA copies can coexist in varying proportions, which is a concept referred to as heteroplasmy. It's very important to also note that while there have been studies conducted in the female germline, healthy adult populations, and mother-offspring pairs, knowledge in heteroplasmy transmission and rate of de novo mutations in the pre-implantation embryo remains lacking, which is due to the restricted availability of donated embryos and the inherent technical bias that's associated with whole genome amplification. So another important aspect of my research is pre-implantation genetic testing. So this is commonly performed with IVF treatment for the selection of a euploid embryo or an embryo that is free from genetic disorders. So during the PGT process, whole genome amplification, or WGA, is necessary in order to obtain enough embryonic DNA collected from a trophectoderm biopsy in order to be able to perform downstream sequencing. And two routinely used amplification platforms are SurePlex, which is used for the pre-implantation genetic testing for aneuploidy, and Repligy, which is used for the pre-implantation genetic testing for monogenic disorders. So the Mitochondrial Genome Project was started by previous master's student Ashi Agarwal at the Create Reproductive Genetics Lab under the supervision of Dr. Librak and Dr. Majunkova. To address the bias introduced by WGA, one experiment involved comparing the two different amplification systems mentioned previously, SurePlex and Repligy. It was found that the Repligy platform provided more uniform coverage when compared to SurePlex, had the highest number of target-aligned reads, the highest percent read enrichment, and the highest mean depth across the mitochondrial genome. This pipeline was then applied to a cohort of 6 familial cases consisting of maternal genomic DNA, embryonic WGA DNA, and placental genomic DNA from miscarriage. So across the 6 cases, there was a total of 23 samples, and a total of 61 heteroplasmies were identified at allele frequency between 5 and 80%. Additionally, it was also found that the average number of heteroplasmies varied based on the sample source, as well as the type of processing, so whether it was unamplified genomic DNA or amplified embryonic DNA. Now moving on to my main aims, my first objective will be to evaluate the mitochondrial DNA heteroplasmy levels and dynamics in the inner cell mass and trophectoderm of human pre-implantation embryos. I will be using 10 embryos donated for research, either aneuploid and donated through biobank consent, or euploid and donated to the biobank after termination of storage. I will also be comparing 3 different amplification systems, SurePlex, Repligy, and Dopplerify, and test which produces the least technical bias while also achieving the highest DNA yield. I will perform mitochondrial DNA enrichment using the twist ligation panel, sequence using NextSeq 550, and analyze the data using Dragon enrichment. My second objective will be to establish the effect that heteroplasmy in pre-implantation embryos has on IVF outcomes, which include ongoing pregnancy, no implantation, and miscarriage. So I will be using biobanked maternal blood or granulosa cells, biobanked placental DNA from miscarriage, and biobanked embryonic DNA obtained from a trophectoderm biopsy. And this will be a retrospective study on selected blastocysts with known IVF outcome. So the third objective will be to study this influence of nuclear gene variants on heteroplasmy levels using the exact same sample set as in objective 2. So for the third objective, the only difference is the type of analysis that is being performed. It's important for research projects to be viewed and assessed from other disciplines so that a broader understanding and appreciation of the findings is obtained. To achieve our goals, the Department of Physiology and the Department of Laboratory Medicine and Pathobiology will collaborate for this project. From the physiological perspective, we will be gaining an understanding in the transmission and distribution of heteroplasmy in the pre-implantation embryo, as well as how nuclear gene variants can affect mitochondrial heteroplasmy levels. And once we understand the mechanisms behind this, we can then use this novel knowledge and implement the expertise of the Laboratory Medicine and Pathobiology Department to create an advanced PGT test. So combining this knowledge and expertise will have a significant impact in implementing mitochondrial DNA variant screening to improve the selection of developmentally more competent euploid embryos and significantly improve clinical outcome success for patients undergoing IVF treatment. One of the most important aspects of research is to ensure that the rationale and the main findings are defined and communicated in an effective way to the public, especially to the individuals that the research can potentially impact. With this project, a patient engagement strategy we intend to use is to create helpful, informational, and easy to understand infographics. So more detailed information regarding the project's objectives, methods, and wider implications will be included, and these infographics will be placed around the clinic so that patients coming in for their appointments can have a chance to look through them and take them home if interested. We also intend to use social media. One of the most effective ways to communicate information is through easy-to-read, quick, and accessible platforms such as Instagram. Our reproductive genetics lab has actually set up an Instagram page where we plan to introduce the project, post updates as it progresses, and show a behind-the-scenes perspective. And implementing a social media strategy can also help to increase the connections between individuals struggling with the same conditions and provide a safe space for meaningful dialogue. And the main goals with these two strategies is to create interactive and open patient engagement to effectively communicate the significance of the science and disseminate important findings in an effective and impactful way. And with that, I would like to take a moment to thank Dr. Librak and Dr. Majunkova on their exceptional supervision, Ashi Agarwal for her significant contributions to this project, everyone at the Create Reproductive Genetics Lab and Create Fertility Center, as well as my amazing committee team. And again, a huge thank you to MITO2i and the Thomas Zackel Scholars for this incredible opportunity. Thank you so much.